Hello flowers. So myself Sanjay Malik and I love flow cytometry. So I'll be talking about flow cytometry and this is a part of a series of videos and you can say that it's part one. It's part one of the series of video where I'll be talking about what a flow cytometer can tell us about and why do we need to do flow cytometry. So first of all, why flow? What is flow cytometry? Flow cytometry, as the terminology, as the term says itself, a flow. So there is a flow component. Cyto cells, metry is measurement. So flow cytometry is basically a technique that measures that makes multiple visual measurements of cells as they are flowing through something. Okay, I'll put it like that, through something. So it's basically a tool or technique to study, as the term says, cells. Okay, so is it that flow cytometry about only cells? No, flow cytometry is not about cells. We can study any particle. Okay, so flow cytometry, by flow cytometry we can study particles, and cells are particles in nature. So whenever we think of then there are many other techniques also. So to study cells or particles, flow cytometry is not the only techniques. So there are many other techniques also. One of the technique being microscopy. Okay. So microscopy is over 250 years old technique, and microscopes are much more affordable. There are microscopes in almost all different educational institutes. And then why do flow cytometry? What flow cytometry? What are the strengths of flow cytometry? How it differs from the current cell analysis methods? So we should not be using flow cytometry just because it can be. We should be having enough justice. We should do enough justice to the flow cytometry as a technology to be incorporated into our studies. Okay, so when do we use, okay, so when do we use method? What is the like whenever we are doing an experiment, we use certain tools, and the tools we use to address certain questions. Okay, so we have some questions. Okay, so we have some questions, and in order to get the answer for that question, we use some techniques. So what are the questions that we can put to a flow cytometer? What the answers that we can expect from a flow cytometer? That's what it would be important. So let's start with, so the first benefit of flow cytometer, or the first strength of flow cytometer, is it can study particles of various different origin. Okay, so it can study wide range of particles. So the particle range could be very wide range of particles. It can study a very wide range of particles. Then two, then it can study multiple different parameters. It can give me information about multiple different physical parameters of a cell or a particle. Okay, so basically it is multi-parametric. Okay. So to just give you an example, in current day flow cytometer, it could study up to 30 different parameters that's commercially available. However, that's not the upper limit. There is still development going on. The flow cytometer are still continuously evolving, and it won't be long that flow cytometer is analyzing more than 100 parameters from a cell population. From each and every cell we have 100 parameters. So what's the benefit of having more parameters? So if you consider, say for example, I put a object. So I'll be talking about very simple analogies. If I look at this from one side, I don't know whether this object is a 3D object or not. I just say it's a planar sheet of paper or something. If I look at it from a different angle, okay, so from a different angle, if I'm looking at it at a different angle, I might see that this is not a square, planar square. This is in fact a tube. Okay, so that means I got to know. So that in order to get this view, I have to see from a different angle. Okay. Now, if it happens that means now the cube has got how many sides? 1, 2, 3. The below surface 4, 5, 6. So the cube has got. So here I can say that 4, 5, and behind it is 6. So the cube has got 6 sides. If the cube is painted with different color, okay, if the cube is painted, the different face of the cube is painted with different color. How from this angle I could only see the cube is having 3 colors. Okay, so from this angle I would have the cube is painted with 3 colors only. If I don't look at the other angles, I don't see. So that means basically I have 100. I have 100 cubes. Okay, and all the cubes are having different combination of these colors, and I cannot segregate or I cannot classify the cubes on the basis of their colors. If I have to classify the cubes on the basis of the color combination, then I have to see the cubes from all different angles. That's where it's talking about multi. So here I'm looking at the same object from multiple different angles. The same way, if I'm looking at the cell from multiple different parameters, that's what gives me much more in-depth information about the subject, about the topic. So this is the benefit of the flow cytometer. It's multi-parametric and it gives me lot of information. So we'll talk about how that lot of information. Third is a flow cytometer can analyze cells at a very high rate. That means the number of cells per unit time. Okay, it could be second or minute per unit time. So if we consider about microscopy, I'll be giving microscopy because that is another cytometry technique. So if I'm talking about cytometry with a regular microscope, it is then with a regular like light field, bright field or fluorescence microscope. In that regular microscope, we could study, generally the study they involve 100, 200 cells. If there is a scanning mode available with a montage or stitching features, with that, with the high throughput screening features, with those features we could see cells say 1000 to 5000. Okay, 1000 to 5000, that we can do with a high throughput screening microscope. It's not a normal microscope, and a high throughput screening microscope with respect to expense, it could be as expensive as a flow cytometer, or it is, and in most of the cases it is much more expensive than a flow cytometer. Say, considering the cost now, since the cost is coming up here, in order to study the large number of cells, say for example, the number of cells per unit time that a flow cytometer can do, 5000 is no way it's comparable to that. Flow cytometer can do 50,000, 100,000, millions of cells. Okay, compared to the microscope, if we put the microscope on a long time screening. So when we compare the number of cells studied by a flow cytometer, so the flow and the microscope, the number of cells studied by a flow cytometer, if it is 1 unit of time, the microscope would take a very high, say for example 10 unit of time to do the similar work. Okay, this is what is the speed of flow cytometer is. And definitely with the microscopy, again the number of parameters that becomes limiting. With the most advanced flow with the most advanced microscopes, now we study 5 to maximum 6. Generally people study 3 to 4, a good, I'm talking about advanced users, 3 to 4 simultaneous parameters they study, whereas on advanced, the much advanced instrument that can do 5, 6. However, flow cytometry is the current widely used flow cytometer, that simple thing that can do 4, 5, 6, 30 is already there in the market. Now, if it is about the cells, then the fourth is we could, when we study large number of cells, what we can detect there is we can detect a rare population. Okay. So what do we mean by rare population is something that is low in prevalence. So say for example, I have a population that is 1%. That means I am getting 1 cells out of every 100 cells. So 1 cell out of every 100 cells. So this is how If I have a population that is 0.1, that is one cell out of every 1,000 cells, this is simple mathematics. So that means now considering microscopy, I'm considering the microscopy here or any other high throughput microscopy method. If we have a population of 0.01%, that is one cell in every 10,000. That means in order to get one cell, I have to study 10,000 cells. In order to get a statistically significant data, statistically robust data, this one cell would not be, is never enough to get a statistical significance. So going by the statistics, what they say we need to have at least 200 events of interest. Okay, so the 200 events of interest, if it is in order to get 200 cells of this 0.01%, consider what is the number of cells we are going to do. If we are going to do two, so this is the number of cells. Okay, so it is 2 million cells. That you consider doing a 2 million cells analysis by the, no matter how advanced the or high throughput microscope it is nowhere happening very soon. And consider if we are having tens or hundreds of samples to be analyzed in a kind of drug screening method, if we are going to do this level of analysis, it's not simply happening. That means flow cytometry has advantages. So here these are the things that it can do. It has a wide range capability. We can study many different particles. It is doing multi-parametric analysis. It tells a lot of different things about the cell. The number of cells we can study per unit time is huge. Because of that, we can study their population. And over and above that, flow cytometry shorters, the flow cytometry shorter capability. They can not only analyze the cell, they can sort and separate the cell population first. So we can say that what is the cell population that we are interested in, and it can give us in a pure form what is the pure cell sorted population. So to make it clear, you have to state it with bold with that all flow cytometers are not sorters. Most of the flow cytometers installed in out there in the academic industries or whatever academics or industries, majority of the flow cytometer are actually analyzers. Very with respect to that, very low percentages sorters. So sorters are generally expensive, and sorting requirement may not be required every time. But yes, with respect to the technology's capability, we can sort cells by this technology. So not only we can see a small population of cell, we can also separate and isolate that 0.1% population into purity, so that we can do further downstream analysis with respect to its proteomics, genomics, the transcriptome. We can do a lot of work. We can culture it further. Then we can do a lot of different studies with that. Then what flow cannot do? We need to know it. It's not that I love flow, so I will talk only what it can do. I'll talk about what flow cannot do. Flow cannot. Definitely what flow cannot. So the one thing is that we get flow with the flow. What we can get is we get only signals. Okay, so we get signals about certain parameters. We don't get any. So since we get the signals, we do not get the morphological information. As I'm comparing a lot with microscopy, we do not get morphological information. So we don't know whether the cell is spindle shaped or it has got a heterogeneous shape. So these are the shape information. To some extent, we can make a calculated guess, or we can approximate. We can get some approximation of the morphology by flow cytometry, but the absolute morphology of the flow cytometry will not be able to. To give you an example, how the relative or how an approximate morphology analysis by flow cytometry has been done is with respect to studies with RBC. Okay. So RBCs have got a very peculiar morphology, that is the biconcave discoid nature. Okay. And whenever there is any perturbation to the RBC it loses its biconcave the discoid form and it becomes spherical. That's where it becomes, be it spirocytes or echinocytes, acanthocytes, whatever is this, the teardrop, whatever is this. In this, the morphology of the RBC changes, and it's a huge change in morphology. So this kind of morphology change could be done by flow cytometry. However, whenever there are very low, the degree of difference in the morphology is low, by flow cytometry will not be able to discriminate that. So yes, morphological information. Then the motility information. Okay. So by flow cytometry, we cannot see if there is any cell movement happening, that we cannot see. By flow cytometry again, so and the other thing that flow does, flow can only see cells or particles in suspension. Cell suspension is only it can see. If we have a tissue, okay, so if we have a tissue, be it liver tissue, so the cells are there in the tissue. In such case, we cannot run it directly on a flow cytometer. What we have to do is basically get the cells in suspension. Okay, so in order to do that, we disintegrate it, or we do a tissue disaggregation using enzymatic method or physical method, chemical method, whatever we do. We have to get the cells in suspension to be able to run on a flow cytometer. By doing so, what we do is we lose the tissue information. Okay? So the three dimensions are the structural information of the tissue, that is what we just get only the cellular information. However, the structural information or the so those are the things that is missing. Okay? So we cannot get that by flow cytometry. So, however, then we will be using flow cytometry only to see what questions that it can address to. And since the flow cytometry can do so many things, it comes with additional responsibility. Whenever there is something powerful, so power comes with responsibility. Okay? Power equals responsibility. Okay. So power comes with the responsibility. One is the responsibility. Whenever there is responsibility, that means there is, so with power there is a chance of abuse of power. That we can get if we are not careful enough, we will be leading to, or we'll be misled by the technology, or we will get we will not be able to appropriately understand the information. That's where, that means with flow cytometry, yes, the challenges increase is expertise. Okay, so we need to have a good expertise developed to do good flow cytometry experiment. It would require experiment design. It would require experiment troubleshooting, validation. So those are the things that if you want to do a new experiment, that those are the things that has to be done. And this is same for no matter what the technology is. The new experiments would require experiment validation and development, troubleshooting. But with flow cytometry, what I have seen with flow cytometry, that is in order to get proper understanding of that expertise has to be developed, and it's not difficult. Okay, I would say that it's not difficult. It does take some time. It requires dedication, or you need to love. So that's why I say that in order to do flow cytometry, you need to love flow cytometry. You cannot just use flow cytometer as just another technique. That is where it can be a very valuable tool for study. And then when the expertise, the initial expense with flow cytometry, generally the instrument cost is relatively high as compared to other, like say for example microscope. But if you are talking about a high content microscope, then yes, flow cytometry, the instrument cost is not much different there, but instrument cost in general could be, but because that's where small labs may not be able to afford that. But having said that, the small labs can always use the central facility, the shared resource facility, from where they can generate way lot of information than their in-house microscopes in the same period of time, or even very less amount of time. That means you are going to a flow cytometer in a matter of an hour, you'll come back with enough information that it would take you months to generate with the microscope. Yes. The reagents, yes, the general notion of people is that flow cytometry experiments are expensive. It's not really that way expensive. Yes, there are some experiments that would require extensive reagents, that would require proprietary reagents, which would cost high, but not all flow cytometry experiments are expensive. Okay. So there are some flow cytometry. Say, for example, I'm working with a marine sample. In that marine sample, I'm looking at phytoplanktons. The phytoplanktons, they have their inner pigments. So those are the kind of sample where I don't even need to add a single reagent. I can just grab some water from the pond or so any aquatic body, and I can run it on flow cytometry, and I can still get information from that. It's about how I put the question, how I interpret the data. So to summarize, with flow cytometry, yes, it can do a lot of work for us. We need to develop expertise on that. Instrument cost is not really prohibitive. Instrument cost, the individual lab may not be able to afford a instrument, but they can always access the instrument once they have their technical know-how, they know their experiment, they have the level of expertise. They can use a shared research facility. And with respect to cost of the experiments, not all experiments on flow cytometry are expensive. There could be something that is way cheaper than a microscopy experiment with respect to the experiment reagent cost. Okay, so yes, flow cytometry has got ample of reason for it to be included or incorporated into your experiment, into your studies, and these are the things that I think it would help you design your experiment. Yes, any type of cells. To like finally conclude this part of the talk, we can study any kind of cells or particles. Okay, so I'll just elaborate on that. When I say it is cells, we can talk about any origin of cells, be it plant, animal, or microbial origin. Plant, any plant. In animal, any animal, be it human, rat, mouse, goat, sheep, you name it, we can do it. And with respect to microbial also, when we are talking about microbial, we have to look at the size of the cell. In my future talk, I will be talking about what is the limitation of flow cytometry with respect to discriminating the size of the cells. So in that article, but if you are working on microbials or any cells, we need to have a eye on the size of the cells. Then with respect to particles, this could be of any particles like it could be a polystyrene particle, it could be plastic particles of any polymer, plastic particles, magnetic particles, metallic particles that we can study. Okay, so we can study metallic particles also. So as long as it's a particle. So one of the experiments that people use particles and flow cytometry that's regularly used is the bead array. It's a bead array. So bead array is an experiment. Again, this is the beauty. It's not that only cells could be studied by flow cytometry. If we can design an experiment that can like here in this bead array experiment. Okay, so in this array, this is equivalent of ELISA. So bead array experiments are equivalents of ELISA that is used to study the soluble analytes. Okay, so how we study soluble analytes, here it is talking about, it's not just cells, it is pushing it beyond that into soluble analytes. So to study soluble analytes, what we use microspheres. So that's where we get the cells or the particles, microspheres. On that we conjugate antibodies. The antibodies will bind to the analytes, and then we can use a second antibody conjugated to a fluorochrome, and then we can look at the fluorescence coming from these bits. So more the analyte present there, more will be the reporter fluorochrome coming from the bits. So in this way, flow cytometry can not only study particulate or particles, it can also be used to study soluble analytes, provided we have the appropriate reagents. So this all gives a huge room to work with flow cytometry. The versatility of flow cytometry is huge, and I would term flow cytometry as a wonder instrument that way. To my knowledge, at least, there is no other instrument which can do so many different things for answering our questions. Thank you. So I'll continue with the other parts of my video, and it is coming up. Stay tuned.